Hi, and welcome to our next video. This video actually is on osmosis. Sorry, it's just taking me a little bit to get ready. All right, so here's osmosis, and it is part of passive transport. This is the second part. The first part, we kind of focused more on diffusion, Brownian motion, concentration gradient, kind of those vocab. Remember, passive does not require any form of energy. So osmosis is the diffusion of water over a cell membrane. And if the concentrations are not equal on both sides of the membrane, water will move to the lower concentration side, just like what we talked about diffusion, from high concentration to low concentration. And there are three different types of movement with different effects on the cell. And that's what we'll look at here. So the very first type of motion is called a hypertonic solution. And when we look at this, we say that the water concentration is higher in the cell and it's lower water concentration outside the cell. And remember that things diffuse from high concentration to low concentration. And because the inside of the cell has a higher water concentration, the outside has a lower, water will move from high inside the cell to low outside the cell. And as it does it, the cell shrinks. And that's the picture there I have up there is kind of a shrunken cell. And that's because water is moved out because of osmosis. Now, these cells that I have here are representing erythrocytes, which are your red blood cells. And you can see that they've shrank a little bit. So maybe this might be putting red blood cells in like a salt or sugar solution where a lot of water is moving outside the cells. You can kind of see, yet again, here's the exact same picture. Hypertonic, the prefix hyper means above. And so if we talk about the water concentration inside the cell is above. And so water is transported outside the cell, high concentration to low concentration. Okay, the next one is called hypotonic. The prefix hypo means below. And so this is where water concentrations are less inside the cell and actually are higher outside the cell. And so what water does, remember, high concentration to low, so it's going to move from outside the cell into the cell. So it's going to go from high to low. And when it does that, we call this hypotonic. Water will move in and it will cause the cell to swell up. And if this would continue unregulated, the cell would actually explode. It would burst because it can't hold any more water. Now, there are some things that do take place to kind of help the cell out so it does not do that. So if you're a plant cell wall will protect you from getting too big. Contractile vacuoles are something we're going to talk about here in a little bit that some cells will have. Or also cells will get rid of some of their dissolved particles. And so there's a variety of different ways that they can do this. And you can see the picture up above. And this is of a plant cell. And you can see that it's no longer a square shaped. It's really kind of rounded now. And that's because there's so much water in it. And that center part right here is that central vacuole. And it's just holding so much water almost to the point that the cell's about ready to burst. So hypotonic, inside less water, outside more water. Remember, from high concentration to low. So the water's going to move from the outside into the cell. The cells are going to get big or possibly burst. The third type of motion is called isotonic, and the prefix iso means same. And this is where concentrations are equal on both sides of the cell. And so what you have is movement out of the cell is equal to the movement inside the cell. So they're going at the same rate. And this cell will not change size at all. And here's a picture of isotonic. So you can see here are my erythrocytes, and they're staying the same size. Red blood cells do have kind of like this donut or kind of lifesaver-like appearance to them. So that is like their normal shape that they will have. So same amount in as same amount out is iso. Iso means same. That's when There's the equal amount of water concentration on either side of the cell. Here, yet again, if we look at with a plant cell, I'm going to go with the isotonic because this is what the cell should normally look like. And so I have equal amounts of water moving out as I do in. And what they do is they represent in with a yellow arrow and out with a green arrow. If I go to the one before that, this is hyper. Hyper means above. That means there's a lot more water inside the cell than outside. So the water is moving out. And you can see that the cell kind of kept its same shape, but the contents, like the cytoplasm, the vacuole, really, really shrank to this. And this would be true with a plant cell because it doesn't have much chance to change its shape. We can see with the red blood cell that we saw earlier, it really did shrink. The last one is hypotonic, and this is where hypo means below. So inside the cell has less water. So water on the outside is rushing in to replace it, and the cell's going to get really, really big. Again, with red blood cells, isotonic, donut-like shape, hypertonic. Hyper means above, so more water on the inside, so it's moving out, so it kind of shrinks. And then hypo means below, so less water on the inside, and so all this water is moving in, and it really swells up. Neither one of the hyper and the hypotonic actually look like donut shape. I know this was a lab that I did when I was in college, real simple, and we used our own blood cells. But yeah, we took like distilled water, we took salt water, and I'm not sure what we did for the isotonic, but real simple, and cells will do this really, really easy. All right, we're going to go on to our video. And since the last time the video did not work, I will be reading this stuff to you. I apologize. So we're going to get this going. And it says diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules such as oxygen and carbon dioxide to cross the plasma membrane. At diffusion, molecules are constantly in motion, they're moving around. Most polymer molecules such as sugar and protein do not really cross this lipid membrane. The water molecules, they're small enough, so if they're small, they can pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. Okay, so we had osmosis going on. Now we're going to add something. So they're adding urea. So if a molecule such as urea is added to one side of the membrane, it will not be able to diffuse across the membrane because it is both large and polar. So you can see that the urea, the green dots, cannot move across. Because of its polar nature, this will interact with the other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. So we should be noticing a shift of molecules between the sides. And with fewer free molecules on the right-hand side, there's now a net movement of water molecules down the concentration gradient to the side with urea molecules. And this is because there isn't as many water molecules to go across. Now look at what happened to the water. As the water molecules are moving to this area, then leaving, the water level on the right-hand side will rise. The water on the left did go down. If the osmotic concentration are isotonic, so we're not going to have any. However, when the solutions have an unequal osmotic concentrations, the solutions with higher concentration solutes, hypotonic, the solution with lower concentration of is hypotonic. So just kind of a little vocab, kind of going back with that little lesson. All right, we're about done with this. I did mention a contractile vacuole, and this it does exist in microorganisms that don't have much control. So this is their way to get rid of excess water. And what it does is it collects water, and then what it'll do is contract and squeeze out some of that extra water. And you can see that here is a microorganism right here, and here are their contractile vacuoles. And so this is how they're able to get rid of extra water. All right. So we got another drawing. You're going to draw a series of images that will explain your understandings of osmosis. You may use an example that you have seen in class or in your book as a reference point. Show the number of molecules on each side of the cell membrane and how it will change over time. And what I mean by molecules is you need to show the water molecules. And I would like to see like what would happen before and after with a hypotonic, a hypertonic, and an isotonic. And look at, here's the end of your slide. See, you survived the second set of notes. And I just need to get this to end.